Welcome everybody. I'm excited to share with you the work I've done as a part of my PhD conducted by Dori and Genela in collaboration with the lab of Adas and also with Professor Vijay Balasubramanian. Got it right, Dori? Subramanian. From the University of Pennsylvania. I'm not sure if he's on the Zoom, but his team is part of our team. So this title actually reflects two separate projects I've worked on. The first was already published in late 2023 and focused on flexible coding of place and time cells in the hippocampus. It was just like an introductory project when I came to the lab. But today I'll be focusing on the second project, which is still unpublished and explores how spatial representations transform across different environments. So the field we are focusing on is navigation. Navigation is fundamental to survival. Animals need to find food, avoid predators, and return to shelter. These aren't just reactive behaviors, they rely on something deeper, an internal model of space, a representation that allows flexible planning and decision-making in dynamic environments. This idea was formalized as cognitive map by Edward Tolman in 1948, long before we had the tools to explore it on the neural level. So in his classic experiment, Tolman trained rats to run through a maze to find food using a specific path, as shown here on the left. Once the rats had learned the route, Tolman modified the maze, blocking the usual path and opening several new arms, radiating outwards, as shown on the right. Remarkably, many rats chose the new path that pointed directly towards the food, even though they have never taken it before. This behavior suggested that the rats weren't simply following memorized turns or stimulus response chains. Instead, they had formed an internal model of the environment, a cognitive map. This idea became a foundational concept in our understanding of spatial representation in brain. So in the decades since Tolman's work, neuroscientists have identified multiple brain regions involved in spatial navigation, including the hippocampus, the medial entorhinal cortex, the subiculum, and others. Within these regions, a rich variety of spatially tuned neurons have been discovered, such as the place cells that activate at specific locations, head direction cells, which are like an internal compass of the animal, border cells that react to boundaries and walls, grid cells, which modulate their firing based on how, fire in periodic grid-like pattern across space, and speed cells, not shown here, that modulates the animal speed. Much of the field is focused on understanding how these cells encode space, both at the level of individual neurons and population dynamics. In our original goal for this project, rather than focusing on the function of specific cells, we asked whether we could use neural activity, the spikes themselves, to reconstruct the environment. In other words, can we build the map from the activity rather than use the map to explain the activity? This concept is similar to how robots use SLAM, that's my background in robotics, simultaneous localization and mapping, to build the map of an unknown environment while simultaneously determining their position within it. SLAM systems rely on sensor data, like LiDARs, which are rangefinders, and cameras, to infer both geometry and location in real time. We propose that the brain may be doing something similar. It uses neural activity from populations of spatially tuned cells to track both the animal's position and the structure of its surrounding, essentially performing a kind of a biological SLAM. The hippocampus formation is widely considered as the center of memory and spatial navigation. It receives processed sensory information inputs and helps generate an internal model of space, enabling flexible navigation and context-dependent memory retrieval. In our study, we focused on two key regions, the medial entorhinal cortex, the MEC, and the subiculum. The MEC located upstream of the hippocampus contains diverse spatially tuned cells, including grid cells, head direction cells, and border cells, which together provide a structured metric-like representation of the environment. The subiculum located downstream CA1 is the main output structure of the hippocampus and plays a key integrative role and projects to both cortical and subcortical areas. Notably, the subiculum is rich in boundary vector cells and exhibits geometric organization in its firing patterns. Since our goal is to understand how the brain might reconstruct the shape and structure of the environment from neural activity, these two regions, the MEC and subiculum, are particularly compelling candidates for investigation. The experiment we planned involved irregularly shaped arenas. So we needed a setup that allows mice to move freely and safely. So here comes my engineering background. I was concerned that because the animals are connected to the wire would get tangled with the corners and the curved walls. So to solve this, I designed a custom motorized tracking system that keeps the tether, the wire, suspended vertically above the mouse at all times. What started as a technical fix actually became a key feature. By applying a gentle directional tension through the tether, we could guide the mouse along specific paths, achieving even and dense coverage of the entire arena. It's like walking a dog. This is especially important for decoding neural activity. A well-distributed trajectory substantially improves the decoding resolution. And it's even more critical in calcium imaging, where there is limited recording time due to the photoglitching effect. We began by using calcium imaging to record neural activity in CA1 as mice investigate a linear track. To validate our motorized tethering system, we compared neural recordings during free behavior and those collected in the guided tethered conditions. The plots you see here are from a tethered session, and they are raster activity maps, with each row representing the activity of a different neuron over time. Color indicates the firing rate, so bright spots correspond to higher activity related to the spikes. Cells are ordered by the onset of their activity, meaning each diagonal that you can see here is actually a span of the entire. Each diagonal here is actually a round trip in the linear track, and what you see here are 5 back and forth trips. Interestingly, the tethered condition revealed place cell sequences very clearly, likely due to the animal's consistent trajectory and speed. While not shown here, we confirmed that the same cells were active across both conditions with no evidence of remapping. Next, we extended our recordings to the dorsal subiculum, which you can see here in orange. Is it orange? Yeah. On the atlas map, the rectangle marks the position of the lens we implanted, which is the window through the miniscope camera records neural activity during behavior. On the left, you can see the matching histological section confirming the lens placement and the recorded area. So we tested the mice in complex arenas, irregularly shaped arenas, and observed robust spatial modulations in the recorded cells, including a population of clear border cells. On the left, you can see the animal's trajectory in black, with the yellow dots marking locations where the specific cell fired. This showed the tight spatial selectivity along the arena's boundary. On the right, we show the activity in the form of receptive fields of the cell, derived from the rate map, clearly aligned with the walls. Here too, we compared free foraging with guided movement and found that the same cells remain active and behave similarly under both conditions. So this is the same cell, once with free foraging, one tethered, and you can see that the spikes happens in similar areas despite the different trajectory. not motorized, not tethered, but not motorized. We then took the first step towards neural-based reconstruction by designing a decoder that attempts to infer the shape of the arena, specifically the position of the walls based on the animal's location and the firing rate maps of allocentric border cells. Just to clarify, allocentric means the cells are tuned to world-based coordinates, like north, what is north, and west and east to the animal, while when we say egocentric, we refer to the position relative to the animal, right, left, forward, and back. What you see here is that the animal explores more, as the animal explores more of the arena, the decoder gradually reconstructs the walls of the environment. This process resembles the SLAM that I've showed before, where the map unfolds progressively over time. But this approach comes with a significant caveat. We are feeding the decoder both with the animal's exact location and the pre-computed firing fields of each cell. So while it shows that reconstruction is possible, it's not too surprising, it's more of a validation step. The real challenge will be to learn the neural activity patterns in one arena and then use them to reconstruct a completely different environment. That's the real test of cognitive map-like representation. At this point, we switched technology. We switched to high-density electrophysiology using neuropixels probes. To adapt this technology, these are advanced silicon-based devices that allows us to record from hundreds of neurons simultaneously with high spatial and temporal precision. To adapt this technology for freely moving mice, we designed a custom protective housing for the probe, along with a precision insertion and extraction tool. These tools enable accurate placement of the probe and safe retraction after the experiment, allowing each probe to be reused once we complete the experiment and move to the next animal. We initially developed this system to NeuroPixel 1 with a single shank and later adapted it to NeuroPixel 2 with four shanks that you can see here. Each shank, what you see here is like a hair. You blow on it and it breaks. So it's very delicate. And this design has been successfully adapted already by 6 labs. The recorded data we processed the recorded data using KiloSort 2.5. It's a spike sorting algorithm that detects and isolates single neuron activity from the multi-channel signals. We then curated the resulting units using the bomb cell tool to ensure high quality data. On average, we retained between 100 and 150 well-isolated neurons per session, yielding rich database suitable for both spatial coding and population-level analysis. In this slide, we show the anatomical targeting plan for the neuropixel recording. As mentioned earlier, we focused on 2 key regions involved in spatial coding, the MEC and the subiculum. In the MEC, our primary targets were layers 1, 2, 3, which include inputs from higher order association cortices and house the somas of head direction and border cells. These layers are also contribute to the superficial input pathway that project to the hippocampus, particularly to the dentate gyros and CA1. In the subiculum, we aim to sample across its proximal distal axis, attempting to capture both the subiculum itself and the adjacent MEC areas. In our recordings, we found, as expected, spatially modulated cells, most notably border cells that you can see here, head direction cells, speed cells, generally spatially cells with the high spatial information. And in the MEC, we saw indications of grid-like activity, but these patterns did not meet the criteria for full classification as grid cells, likely because the arenas weren't large enough to reveal their periodic structure. On the right, you can see a summary of the number of cells we classified in 2 animals. This is from 2 different experiments. Some classifications, as you can see, overlap, either due to classification uncertainty or because certain cells are conjunctive, responding to more than a single spatial feature. So finally, in our experimental paradigm, mice explored two connected rooms, each with a distinct geometry. The session begins with 20 minutes of recording in room A. Then the animal was transferred through a doorway to room B for 20 minutes. And finally, it was returned to room A, we call now A prime, for another 20 minutes. This A prime structure allows us to study how spatial representations transform between environments and to use A prime as within room control for stability and consistency. So here's a video shown at 4 times speed, capturing the experiment from the two perspectives, the top camera and the mobile camera that follows the mice. And in a moment, you will see Gaia opening the door for Alex, the mice, to move from room A to room B. We had various room combinations used in our experiments, some with symmetrical relationships between the rooms and others without. We worked with 6 animals, 3 recorded in the subiculum and 3 in other regions, including the MEC and pre-parasubiculum. Altogether, we conducted 80 trials and recorded over 6,000 high-quality units as validated by BOMCell and more than 10,000 units classified as good with kilosols. So here are examples of border cells, firing rate maps of border cells, analyzed separately in room A and room B, but displayed together just for comparison. You can see that the receptive fields of these cells exhibit different transformation patterns between the rooms. Some maintain their allocentric orientation. Some are mirrored across the axis, usually the bisecting wall. And others show a rotation. This example here illustrates the stability and transformation of firing maps across rooms. Each column is a different cell, and the rows are showing the activity in room A, B, and A prime. There is a rather consistent activity patterns in room A, and it's repeated room A prime, demonstrating within-room stability after visiting room B in between. However, the firing map in room B is different than room A for many cells. The observation that some cells change their encoding between the rooms led us to realize that reconstructing room B based solely on neural activity learned in room A is inherently challenging, unless we first understand the nature of the transformation. Moreover, while cell-to-cell analysis is informative, the more powerful and biological plausible approach is to examine the population-level activity, since the brain likely operates using distributed codes across neural ensembles. So to assess the correspondences of population activities between the environments, we compared firing rate vectors across spatial bins. For each bin in room A, we constructed a population vector, a vector where each element represents the firing rate of a different cell at that location, here shown for 3 cells. We then correlated this vector with all population vectors from room B, identifying the bin in room B with the highest correlation. If this correlation was significantly higher than the correlations to the other bins, we consider it a matched bin. This yields the mapping between spatial bins of room A and room B, based purely on similarity in the neural population activity. To illustrate how spatial bins in room A corresponds to those in room B, we use the gradient color map applied along the x-axis and the y-axis of room A. Each bin in room B is then colored according to its most correlated bin in room A, based on the population vector similarity. This animation shows how room B is painted using room's A gradient, revealing the underlying transformation if exists, in this case, a rotation and shear. This is just an emulation. By visualizing X and Y mapping separately, we can intuitively observe whether the neural representation involves a rotation, mirroring, or other systematic changes. Before diving into the results, let's take a moment to introduce affine transformation. since it's central to our analysis. An affine transformation is a mathematical operation that reshapes space while preserving structure. It keeps straight lines straight, maintains parallelism, and preserves ratios of distances along those lines. However, it still allows for shifts, rotation, scaling, and shearing. For example, this square can turn to a rectangle, or this square can turn to parallelogram. In our case, we're looking of how the population activity in room A is transformed into population activity in room B, and asking if this transformation be described by an affine operation. This includes not just translation, like shifting the entire map, but also changes in angle, scaling along specific directions, and even shearing the structure. So here's an example from one of our recording sessions. What you're seeing are two 3x3 matrices showing a room-to-room comparison. The left matrix visualizes the horizontal transformation, and the right one shows the vertical transformation. Each matrix reflects how neural population vectors from one room maps to the other room. The rows represent the source room, and the columns represent the target rooms. So for example, the diagonal is comparing a room to itself, and naturally, this is the identity transformation, and seen very clearly because it's correlating the data with itself. What's more interesting are the off-diagonal, A to A prime and A prime to A, which you can see resembles the original pattern, which evident on stability. But the more interesting parts are A to B and B to A prime. That's where we try to look at the transformation between different environments. To quantify this, we applied affine regression, fitting the matched spatial beans using a combination of rotation, scaling, and translation. The colored arrows that you see here are orthogonal vectors. And once they go through the regressed affine transformation, they're turned in direction and in size, according to the scaling and the rotation of the transformation found. This gives us a geometric framework for understanding how spatial representation shift across environments. Here you can see a few more examples. The color code transformation maps indicate the relationships between the rooms. Left column examples show a rotation. Here we see mirroring, and the rest seems like no change. So the identity transformation. So affine transformations provide a useful and interpretable model and can be quantified by measuring how much variance in the transformation is explained by comparing the projected points and the actual points. But we also wanted to ask a broader question. Could the transformation between spatial maps be structured in other ways, perhaps in ways that are non-linear, yet still preserve local neighborhood relationships or global smoothness? To explore this, we assembled a set of complementary metrics aimed to detecting whether the transformation is meaningful or whether it simply reflects random noise, like a disorganized salt and pepper pattern. These metrics help us to go beyond visual impression and to start to quantify the structure in the transformation, even in cases where affine model might fall short. First, we look at the mean correlation between matched spatial being across rooms. Higher correlations suggest more structure. Then we examined ambiguity. How many source beam maps to the same destination in room B? A low number of ambiguous mappings indicate that the transformation is informative and well-defined. We also computed the Laplacian smoothness with the courtesy of Hadass, which measures how smoothly the transformations varies across space. Conceptually, it reflects whether neighboring beams in room A remain close together after the transformation to room B. A lower score means a smoother transformation, indicating that local spatial relationships are better preserved. In addition to Laplacian smoothness, we used other metrics such as the number of neighbors maintained, and Earth moving distance, which is another metric that measures the energy taken to take one distribution to the other. Together, these metrics let us go beyond visual inspection, providing a quantitative framework to evaluate how structured and consistent the transformations are, even in the presence of noise. So to determine whether the smooth and structured transformation was observed were truly meaningful or could have emerged by chance, we introduced several shuffling controls. Specifically, we applied different types of randomizations to the population vector rate maps in room B, aiming to break any real correspondence with room A. This included shuffling the order of the cells, shuffling rows and columns, shuffling in 2D, and also circular shuffling aimed to mask the border cells, which tend to be in equidistances from the perimeter. Together, these manipulations allowed us to test whether the transformations we observed generally reflects underlying neural organization, or whether they could be simply artifacts of the analysis. This slide presents results from a single trial where we generated the null distribution by shuffling cells identities, so the number of the cell number in room B. So they are not and not aligned. Each cell in room A now do not correspond to the same cell in room B. And each histogram here shows the distribution of metrics values across 1,000 shuffle rotations. The red vertical line marks the value computed from the actual unshuffled data. And as you can see here, it shows a strong statistical significance. This suggests that the observed transformation is structured and non-random. In contrast, in trials where the transformation is noisy, which resemble the salt and pepper structure, typically they yield metrics that are less distinct from the shuffle distribution, and the statistical significance is lost. In these slides, we show the percentage of trials from the total of 80 trials that we had, in which the transformation between rooms was statistically significant compared to various types of shuffle controls. Each pair of bars represent a different shuffle type, and each line is a different metric. The green bars indicate results from the MEC animals, and blue bars are from the subiculum animals. If you look at the two first rows, which show correlation and ambiguity metrics, we see that about 50% of the trials are significant in both regions. This consistency suggests that the transformations aren't random, but reflect structure change in representation. In the bottom row, showing the Laplacian smoothness metrics, we observed higher proportion of significant transformations in the subiculum compared to the MEC. This could reflect the smoother or more structured transformation in the subiculum, consistent with its role as an output hub of the hippocampus. So the data is more processed and more mature. Overall, these results support the idea that the transformation between environment is robust, reliable, and structured, especially when compared to shuffle baseline. Yep. Thank you, as Doris said, it's an excellent question because I know the answer. In this kind of shuffle, we actually did not mix the cell identities. So they went through the same movement. So it's only a spatially location transformation. Therefore, the correlations are the same. Okay, so they will score the same score in the correlation because we just moved them in the area. At this point, we asked ourselves, can we find a more powerful and flexible way to understand how spatial representations transform between rooms? To explore this, we collaborated with Adas Benishti's lab, and here and Doris' super talented graduate student, Daniel. And they tackled the problem from using deep learning. Specifically, they used LSTM, which is a long short-term memory network. LSTM are a type of recurrent neural network designed to process sequences and capture temporal dynamics. which is ideal here since the animal's neural activity evolves over time as the animal navigates in space. The architecture built here decodes the animal's 2D position from the recorded spiking activity. It starts with a shared LSTM, which received about 4 seconds of spike data from many recorded cells. And this LSTM learns how varying patterns unfold over time and encodes them into the latent representation, essentially compressed summary of temporal activity structure. That shared latent space is then passed into room-specific decoders, one for each room. Each decoder is a small feed-forward network with 4 fully connected layers, transforming the latent representation into 2 outputs, the predicted X and Y locations, coordinates of the animal's position. So here we see a summary of how well the neural network model decoded the animal's position across 3 rooms, the A, B, and A prime. On the y-axis is the prediction score in R square, which is the explained variance, which tells how closely the predicted positions match the actual positions. Each box represents the distribution of prediction scores across sessions for a given room, and the line inside the box is the median. In each dot is a single session. What you can see is that the decoding accuracy is relatively high in all 3 rooms, typically between 60 and 70%, showing that the model performs well regardless of which environment it was decoding. The outliers at the bottom may be due to noisy sessions or fewer recorded cells, but overall these results tell us that the model generalized well and sets the stage for comparing activity between rooms to analyze transformation. So now we can move to the more interesting question. How do we decode positions? How do we find the relationships in positions between the rooms? To do this, we take the neural activity recorded in room A and feed it through the shared LSTM and each of the room-specific decoders. This gives us predicted positions not only in room A, but also in room B and room A prime based on the same activity. In other words, for every moment the animal is in room A, we can estimate where the same neural activity would place it in room B and room A prime. This gives us a set of match points, pairs, like a functional mapping from A to B and from A to A prime. And the same can be done if the activity is in room B and then decoded to room A, and similarly with the room A prime. Once we have these match coordinates, we can perform an affine regression to characterize the transformations and test whether the neural representation transforms a structured geometric way or whether it's more complex and unpredictable. To demonstrate, Daniel prepared us a nice video from real data. So the animation is based on real data and illustrates what we have just described. The black snake shows the animal's actual trajectory, while each color represents the predicted position in a different room, decoded from the same neural activity. So for each moment in time, we decoded where the activity would place the animal, not only in the actual room, but also in the other rooms. And that allowed us to match the activity between the rooms and ask whether there is an affine transformation here or not. This slide summarizes well the affine transformation score, again in R square in explained variance. So each group in the x-axis shows a different room comparison. The control comparison, A to A prime and A prime to A, in the middle shows higher R square scores as expected, meaning that the neural code remains fairly consistent over time when the room stays the same. The cross-room transformations A to B and A prime B have lower scores, but yet substantial. And here's a similar setup to the one I showed earlier for the population vector analysis, showing all room comparisons with horizontal and vertical gradients. Again, you can see here the diagonal is comparing the room to itself, which is obvious, but there is a nice A to A prime and A prime to A correlation. So the maps look almost identical. And the interesting part is A to B and B to A prime, where you can see already a rotation of the arena, but a clear structured, smooth transformation. And here are additional examples that I shown before, but this time with the analysis of the neural network. And you can see that they quite match. This is the population vector, and this is the neural network. And here we compare the transformation angle estimates between the two methods, the population vector correlation and the neural-based matching. The results show a fair agreement when we exclude the mirror transformations, the green ones, that I will discuss later on. Now that these two methods approach the problem very differently. The population vector method uses static firing rate maps. It looks at where each neuron fires on average. In contrast, the neural network method uses the LSTM model that learns from the temporal dynamics of the neural activity, and the fact that both methods, despite these differences, converge on similar transformations, tell us that the transformation is real and robust. This figure, again with the courtesy creative Daniel, shows all trials plotted by their position prediction accuracy in room A, it's the x-axis, versus their affine mapping score from room A to room B, the y-axis. Each trial is represented by an image of the arena, and the colored outline indicates the animal ID. The upper right quadrant represents trials with both strong spatial encoding and strong cross-room mapping, which are likely the most reliable and meaningful. Therefore, we tend to focus on them on the downstream analysis. So let's take a moment to summarize what we've discovered so far. First, we identified allocentric border cells and other spatially tuned cell types in the subiculum complex and MEC. This is not new, giving us a diverse neural population to work with. We observed that cell encoding changed when the animal entered the new room, indicating that the neural representation is context-dependent. Interestingly, we found that the population activity in room B could be described as an affine transformation of the activity in room A, capturing changes in position, angle, and scale. And when we approach this using deep learning model, it converged on similar transformations, reinforcing the robustness of this mapping. At the same time, we saw that rooms A representations remained stable when the animal returned to the same room, suggesting that the transformation wasn't due to noise or drift, but a meaningful shift in representation. So we've established that the transformation is structured and reproducible. But now comes the next question, which we're still exploring. What governs these transformations? One option to consider is that the transformation follows a sensory input. One possibility is that the visible perceived similarity between the rooms cause the map to realign to what the animal interprets as the new orientation, essentially a perceptual anchoring. Another candidate is the use of visual landmarks, such as distinct corners. These may serve as reference points for alignment. And in fact, recent work from the Giacomo lab reported evidence of corner cells in subiculum, which add weight to this idea. Another possibility is that the transformation itself is governed by the animal's behavior. For example, the way it explores each arena. This idea connects to models like the successor representation framework, which propose that the structure of a cognitive map reflects the expected future occupancy, essentially how the animal is likely to move through the environment. In this view, the transformation between neural representation isn't arbitrary. It reflects changes in navigation patterns and how the brain anticipates future states. Supporting this, recent studies have shown that neural coding can align with the structure predicted by successor representations, suggesting a behaviorally driven organization of spatial maps. To understand the source of spatial transformations, we can track how specific type of neurons encode space across conditions. Head direction cells, which act like internal compass, often maintain coherence within systems even with visual cues are rotated or shifted. They can preserve their relative directional tuning despite conflicting environment cues. This makes them a powerful anchor to infer rotation angles between environments. Next, we turn to grid cells. These cells form a hexagonal firing pattern that aligns with the geometry of the space. When the environment is transformed, such as through rotation or stretching, grid cells often realign accordingly. By analyzing these realignments, we can estimate not just whether a transformation occurred, but how it was structured. Together, these encoding patterns help us track the transformation in spatial representations by comparing the cell's activity across environments. So we looked into several structure features of the environment, such as their orientation, symmetry, the geometry of the bisecting wall, but none of them yet have provided a clear, consistent explanation of the observed transformation. However, we did find two intriguing patterns. The first one concerned the scaling factor in the affine transformation. When we decompose the transformation matrix, we notice that one axis typically has scaling factor close to 1, as you can see here, but the scale here starts with 0.4, so it's a bit misleading. So the results are around 1, suggesting no change in that dimension. But the other axis, this one, often has scaling factor less than 1, which implies a kind of compression along the other direction. We further looked at if the transformation matrix scaling axis align with the geometry of the room. To quantify this, we compared the direction of the major scaling axis, the axis with the largest stretch of compression in the affine transform, to the main axis of variance in the room geometry, for example, the long wall or the open direction. What we found is that there is a linear relationship between the two axes. The greater the variance along the environment axis, the more likely it is aligned with the transformation main scaling direction. When we look at the minor scaling axis, the one showing the greater compression, just like with the major axis, we found the linear relationships between the variance in that direction to the axis of the room geometry. This means that both major and minor axes of the affine transformation seems to align with the room's physical dimensions. So although we did not find a consistent explanation based purely on rotation angle, these findings suggest that once the transformation angle is set, perhaps anchored by head direction or cues, the scaling follows the structure of the environment. Our second observation relates to reflections or mirroring in the transformations. Interestingly, these reflections appear exclusively in animals recorded from the subiculum. And in one particular animal, we saw reflections in a high proportion of the trials. Note that we're showing here only trials which pass the R-squared criteria, so it's not noise, it's reliable features. This caught our attention because previous studies have linked the subiculum symmetry to symmetry processing. So there may be a relationship between the presence of reflections and how the subiculum encodes spatial structure. This is something will be interesting to further explore. So to wrap up, we started this project with the goal of reconstructing cognitive maps from neural activity. But as we dove deeper into how activity maps across different arenas, we made an interesting discovery. We found that the population activity maintains structured spatial representation across environments, and that these representations are often related by a systematic affine transformation. What governs this transformation is still open question. We're actively investigating it, what can drive it, including the intriguing mirror transformations we observed in the subiculum. The significance of these findings is that they suggest a predictable geometric logic to how spatial representations are restructured across contexts, rather than being entirely random or arbitrary as we see in CA1. Understanding this transformation could be a key step towards decoding how the brain generalizes spatial knowledge and how it flexibly reuses existing representations to map new environments. And with that, I'd like to take a moment to thank everybody involved in this project. Eli Luria is a close friend, not in the faculty, that helped me develop the real-time part of the tracking system. Sivan Ashkenazi, hopefully, oh, she's not on the Zoom, but she was my mentor when I when I came to the lab, Ohad and Sheila for their assistance on the neuropexil surgeries, Gaia and Shachaf, who became pilots on my experimental setup and ran many of the experiments. And as you can see, also co-pilots. Salman, hopefully on the Zoom, is always ready to help anyone with everything. Daniel from in the DAS, and recently Kesem, who became deeply engaged with the project and major contributors to it. Professor Vijay Balasubramanian and his team for their insightful discussions and creative perspectives. And very special thanks to mom and dad, Dori and Ganela, not only brilliant scientists, but also genuinely kind people and really enjoy to work with. And Dori, you took me, I wanted to tell the story. You already told it, how I got to the lab and how Dori pushed me, say, if you're here, so why not doing a PhD? So I did it despite my gray hair. I'm also thankful to Irena, our lab managers, all our lab members, and the neighboring labs of Yoram, Jackie, Ben, Shai, and Omri, and the students, many of them, or most of them, the age of my kids, and made me feel like one of them, not their dad, despite my daddy jokes. And most importantly, to my amazing family, my wife, Irit, Shachar, my youngest, Maya, who's en route to Mongolia, and my elder boys, Omer and Eran, who are still in service, make sure that we are protected.